Hi students, I have a lecture today on membranes and their fluidity. So I just want to do a little video lecture for you so you can see some of the things that I find important. In this picture we have just a small cross section of the plasma membrane, and it contains phospholipids, shown in red, studded with different proteins. And the phospholipids and proteins make up the bulk of the plasma membrane. There are some other molecules as well found in the membrane and attached to it. We're going to talk about those today. So for one thing, the membrane is a fluid structure. It's not a solid sheet that covers the cell, but it's constantly moving. If you could imagine the surface of a bubble, if you blow bubbles, it's kind of shimmering and you can see that it's in motion. Or the surface of a pool, and you can see that beach balls can float on a pool. That's what the membrane is like. For the structure, we have the phospholipid base, and its main function is to contain the cell's compartment on its own. It's selectively permeable, and some things can cross easily across that membrane, while other things cannot. So please review in your textbook the permeability of different substances. You can see that certain ions will cross the membrane easily and other ones will not. The model of the plasma membrane is often called a fluid mosaic model, where the fluid part of the membrane is the phospholipids, they're in constant motion, and the proteins are the mosaic. Here we go. The fluidity of that membrane is influenced by a variety of factors, especially temperature. Phospholipids are fats, or they're related to fats at least. And when you cool down a lipid, you make it more solid, and it becomes less liquidy, less fluid, more viscous as the temperature decreases. So when temperatures get low, the plasma membrane becomes less fluid. When temperatures rise, just like melting butter or lard or heating up oil, the membrane, the phospholipids become more viscous, very fluid and very liquidy. This is influenced partly by membrane composition. So lipids can be saturated or unsaturated, and unsaturated fats or lipids have little bends to them, which make them more fluid at any temperature, more fluid than saturated fats, which don't have bends and they're straight. So unsaturated fats on the phospholipid membrane will make it more fluid. Another important molecule found in the plasma membrane is cholesterol, shown here and here in yellow. Cholesterol is between some of the phospholipids on the plasma membrane, and this can get in the way of packing those phospholipids. So cholesterol can influence the fluidity of the membrane as well. Mainly when temperature rises, the cholesterol restrains the movement or fluidity of the plasma membrane. So as temperature rises, that cholesterol keeps the membrane from becoming too liquidy. If temperatures decrease, the cholesterol packed in between the phospholipids keeps them from getting too tight and keeps the cell somewhat fluid, even at low temperatures. So cholesterol is kind of a moderator in the fluidity of the membrane. The proteins, as you saw in the first slide, are very important to the cell in the plasma membrane. They're throughout the plasma membrane, and there are different types of membrane proteins. Some of the proteins go throughout the plasma membrane. These are called integral proteins. I can move this, that's interesting. Okay, the peripheral proteins are not embedded throughout the membrane. They are just on the surface. So here in this membrane picture, This would be outside the cell, this would be inside the cell, and this protein molecule right here is partly inside the cell, and it's exposed on the outside as well. So these protein molecules have several functions there, which we're going to discuss. There are 4 major functions of these membrane proteins, and one of them, I think the most important, is transport. These proteins can help move substances across the membrane. They can act as a tunnel, or they can shuttle molecules back and forth. The proteins can also be involved in enzymatic activity, that is, chemical reactions that require enzymes. Many proteins are enzymes, and these can be embedded in the membrane as well. Also, proteins help cells communicate. They can be like ID badges for the cell. So cells can recognize different proteins and respond to other cells. This also feeds into cell-cell recognition. Red blood cells, for instance, have different antigens on their surface, and these antigens can be recognized by other cells. So somebody who has type A blood has type A antigens, and if that blood gets transfused into somebody with type B blood, their cells recognize the antigens and will fight it off. Conversely, if somebody with type A blood donates to somebody with type A blood, the antigens recognize each other and accept each other. So the membrane is this somewhat fluid substance surrounding the cell. It's a sac that holds everything in, the cytoplasm and all the organelles. But the cell interacts with other cells in a lot of different ways and interacts with its surrounding environment. So some things need to come into the cell. Sugars. The cell needs sugars for energy. Water. Ions. Different types of molecules will enter the cell and exit the cell. Things that exit the cell can be hormones or waste products. These things need to go across the membrane to get in or out of the cell. So different substances will do this in different ways. Hydrophobic molecules, those that do not interact with water, they're the more fatty ones. They're, because of nonpolar bonds, not going to interact with water very easily. They can easily, however, transport or diffuse across the plasma membrane and the lipid bilayer. Okay, so a fatty substance or a nonpolar molecule can get dissolved in that fatty part of the phospholipids and cross the membrane. Channel proteins, those that go throughout the cell, sometimes we call them integral proteins, can help substances move across, acting as a hydrophilic tunnel. So molecules that are hydrophilic and tend to have polar covalent bonds can just be transported through the proteins. Carrier proteins can also pick up some hydrophilic substances, and they will take up molecules, change shape, and transport those molecules across the membrane like a ferry, like over there. So those are the integral proteins, and the transport integral proteins can move from one side of the membrane to the other, either bring things into the cell or shuttle substances out of the cell. Okay. Passive transport is diffusion and osmosis. Passive transport refers to types of transport that do not require an input of energy. So while those carrier proteins can move across the membrane, that takes energy in the form of ATP, that's cellular energy. Some things do not require any energy, such as those hydrophobic molecules that can diffuse across the lipid layer. Diffusion is spontaneous. It happens naturally in cells and just in the environment. Diffusion is when substances move along a concentration gradient from an area of high concentration to an area of low concentration. So if somebody walked into the room wearing strong perfume, that perfume, those molecules would diffuse throughout the room, and eventually we could all smell the perfume. That's diffusion. It's any substance going from an area of high to low concentration, and sometimes that's across the membrane, sometimes it's not. Osmosis is a special type of diffusion involving water. Active transport occurs when ATP, or energy, is needed to deliver substances across the membrane. So a good example of this is the sodium-potassium pump. ATP supplies the energy needed to move sodium, its atomic symbol is capital N lowercase a, and potassium, which has the atomic symbol of K, across the concentration gradient, against the concentration gradient. So here we pump sodium to an area of higher concentration, and we pump potassium to an area of higher concentration. The sodium gets pushed out of the cell, the potassium comes into the cell. And there are several steps to this process. In the first step here, the sodium binds to the protein. It's an integral protein going throughout the plasma membrane, and sodium will bind to it. It's open on the inside of the cell. ATP phosphorylates. That means it delivers some of the energy, and a phosphorus of the ATP molecule gets removed. That causes the protein to change shape, and the sodium gets dumped out of the cell. So you can see there are 3 sodium ions that get shuttled out of the cell. Now that protein is exposed to the external environment, and it has binding sites for potassium, 2 binding sites to be exact. So those potassium ions will bond to those sites, and ADP is bonds to a phosphate. So we actually make some ATP here. And then the potassium is delivered into the cell. So you'll see here there are 2 potassiums that go in for every 3 sodiums that go out of the cell. That creates this electrochemical gradient across the cell. We pump out 3 positive ions, 3 positively charged things, and we get 2 positively charged things. So outside of the cell becomes more positive, and the inside of the cell becomes more negative in charge. This is called an electrochemical gradient. It causes a voltage across the membrane. I put a video on our Moodle site showing how this relates to nerve conduction in the human body. The ions create the gradient because they have the charge. Large molecules can enter and exit the cell in a couple different ways. They might have trouble diffusing through the lipid bilayer, or they might be too big to fit through a protein tunnel. So substances that need to exit the cell will go through exocytosis. These are big molecules, maybe big proteins, that leave the Golgi apparatus in a vesicle, and that vesicle, being a phospholipid bilayer itself, will fuse with the plasma membrane, open up, and dump the contents out of the cell. So the key thing to remember here is exo, exiting. Endocytosis is when substances enter the cell. Endo means into or entering, and here the cell takes in macromolecules. This picture shows molecules outside the cell, and when they come in contact with the plasma membrane, a vesicle forms at the membrane and brings those molecules into the cell. Endocytosis. Cyto means cell. There are a couple different types of this. Pinocytosis is when a cell engulfs fluid and also macromolecules. Phagocytosis is called cell eating. Phago refers to eating, and here the cell engulfs a food molecule with structures like pseudopodia. We see this in lab when we were looking at the amoeba. They have those projections of the plasma membrane right here that can wrap around food particles. The pseudopods can be used for moving the amoeba around, but can also be used for consumption of large molecules and little bacteria. Receptor-mediated endocytosis is a type of endocytosis where we form these concentrated vesicles. So there are receptors on the surface of the cell, certain cells, that will pick up only certain molecules. And so these receptor sites can grab the molecules that they need and form a concentrated vesicle, bringing that concentrated substance into the cell. Okay, I hope this helps in your study of the membrane. We're going to be looking at lipids, proteins, carbohydrates, and nucleic acids very soon. So enjoy.